It is my great pleasure to present our plenary speaker today, Stephan Gekle, who is a professor of theoretical physics at the University of Bayreuth. There, he develops top research using both numerical simulations and physical modeling with the purpose of describing many different types of processes, such as the flow and the formation of real blood cells in blood vessels, the supramolecular aggregation in simple solvents, this performed using MD simulations, the construction of tissue structures by 3D printing of living cells, or the transport of microplastics from the ocean to the atmosphere. Today, he will be presenting the talk entitled Simulation of Blood Flow as a Multiphase Fluid. Please, Stephan.
Yeah, thank you very much, Jose. Thank you very much to the organizers for inviting me to give me the opportunity to present. So I'm going to talk about simulations of blood flow with the perspective of the multiphase fluid, because that's what fits to the workshop. If that jumps, yeah. So let me start by some introductions, some motivation of why blood flow is a multiphase fluid. I'm just going to show you some snapshots of simulations that we're doing, and then we'll focus on one part of it. So this is a typical situation of how you would probably imagine blood flow to look like. The red things are obviously the red blood cells, and the green things here are other cells or artificial particles or The main ingredient is the red blood cells. In fact, in large vessels, the volume fraction of red blood cells is between 40 and 50%. So blood is really a very dense suspension of cells, and these red blood cells, as you can also see from the movie, are highly deformable objects. So they're not spheres, and they are also not rigid biconcave discs. So the typical picture that you may have in mind of a red blood cell is this biconcave disc shape. That's what the shape is if the cell is not moving, but as soon as the cell starts moving in the flow, it heavily deforms and, well, looks in the end like this suspension here. Another aspect then to understand such things is to focus on a single cell. So that is an example here. That is a channel, and the camera, you have to imagine the camera is moving with the red blood cell, and the flow here is in fact perfectly steady, so the externally imposed flow, but the cell nevertheless does these permanent rotations, does these permanent oscillations, so it actually breaks the symmetry. So even for a single cell, there are many, many very interesting effects in these red blood cells. But there's not only red blood cells in blood flow. Another aspect that one could look at is the formation of thrombi that is formed by platelets. Platelets are very small cells. They're actually not deformable. And in this simulation that I'm showing here, there are no red blood cells, so these things are the platelets, and the platelets can adhere. So the platelets are sticky, and they can adhere to the surface, and then they can form these things that eventually will grow into a block. Also here, one can look at not only the collective aspects, but one can also look at a single cell, single of these platelets, and here especially the formation of these platelets. So these cells need to be produced constantly in our bodies, and one can ask, how does this mechanism work? How are these cells being produced? And how is this production maybe connected to flow, to a multiphase flow? And in fact, what I'm going to show you here is the platelet formation, which is connected to interfacial instabilities. So in the last years, we mainly worked on the top two aspects, these two. But then when I looked at the program of this workshop, I figured that probably most of you will be most interested in this latter part here, in these interfacial instabilities. So what we're going to do is I'm going to introduce the simulation method, focusing on cell models in general, red blood cells, but also for other cells. And then we'll go more into detail on this lower right aspect of the platelet formation and how this is connected to an interface and how this is connected to an interfacial instability. So the outline is that first the methods, I'll be speaking about the model for the cell. So we need a mechanical model for the cell, and about the model for the solver, where we're going to use a lattice-Boltzmann fluid solver. Then we're going to look at the formation, or biogenesis if you want, for platelets. And then finally, I'm going to show you how this is connected. I'm going to do some theory, how this is connected to, or we think it is connected to, a Rayleigh plateau instability with some special ingredients, namely some anisotropy. Right. So here's the red blood cell if it doesn't move. It has this disc shape. It's about 8 micrometers in diameter. And we describe it with a triangulation. So we have these triangles, vertices are connected by triangles, and these describe the surface. A red blood cell is essentially a bag of liquid. So there's the plasma outside the cell, and there's liquid inside, and the membrane separates these two compartments. The inner fluid is a solution of hemoglobin. That's the protein that transports the oxygen. So that's the essential function of red blood cells. The viscosity ratio, so the ratio between the viscosities inside and outside, is called lambda. It's about somewhere between 5 and 10. The exact value is not really known, but in that range. So the inner part of the cell is more viscous, and the inner part, as I said, is liquid. So that distinguishes the red blood cell from many other cells. Many other cells, they have a nucleus, they have a cytoskeleton, they really have an elastic, a solid structure. Inside, red blood cells not. They have a membrane, and inside there's basically just liquid. They don't have a nucleus, so they expel the nucleus when they are being produced. The membrane structure is very complicated. It has hundreds of different proteins, but it has two main components, which mechanically are the most important components. And the first one is the lipid bilayer. So that's a layer here formed of these lipid molecules. And below that is a network of proteins, an elastic network of proteins, mainly these so-called spectrin proteins. The total membrane thickness is about 100 nanometers. If you compare that to the 8 micrometer diameter, you can consider the membrane as thin, and that's in fact what we do in our models. So what we'll use is a 2D elastic sheet, which describes our membrane, which has no thickness. In the simulation, it doesn't have a thickness. Such a sheet has 3 deformation modes. 2 of them also exist for a 3D solid. The first one is shearing deformation, and the second one is a stretching or area deformation mode. And the third one, which has no exact equivalent in a 3D solid, is a bending. So you can take the membrane and you can bend it. And in fact, there's a resistance also to bending. So the membrane resists all these deformations. So what we need is mechanical models to describe these resisting forces. Since this membrane is a complicated structure, the approach is to use a phenomenological approach that has been pioneered by Skalak in the 1970s. So he did basically measurements, simple measurements on the mechanics of these red blood cells, and then he wrote down an energy functional describing these forces that he saw without any direct connection, any direct link to the microscopic structure. So phenomenological energy. This is what it looks like. These I's here are the invariants of the deformation tensor, and this W is the energy density. It has 2 moduli, so it has 2 phenomenological parameters. One is the shear modulus, kappa S, and the other is an area dilation modulus, kappa A. These parameters are very difficult to obtain because isolating a red blood cell and then really doing isolated experiments on these red blood cells is difficult. Also, the medium depends a lot. So you have variations between the different cells, but some range of values are known, and that is, did I write? Oh, I didn't write it. So the kappa S has about 5 newtons per meter, and the kappa A, the area dilation, is much larger, which means that red blood cells basically cannot extend the area. So the area is a pretty well conserved quantity.
The area is conserved, but not the volume, correct?
The volume is also mainly conserved, but they are initially not a sphere. That's why they can deform. Because the initial shape is this, and that's why they can still deform, even though they are conserving area and volume. So they're still well, the area, yes, that is relatively well known. It is also quite well known that the volume is conserved as long as you don't change the medium. If you put salt in the medium, that changes because of osmotic pressures. And there may also be some active effects. So it's not fully clear whether the membrane is really tight or whether there are some active mechanisms by which the cell regulates the volume, so water pumps. So that's an issue that is at the moment not fully known. So for us, the volume. So we conserve the volume. And the next component is the bending resistance. The approach here is the same, so we write down a phenomenological energy density, or a person called Helfrich wrote it down again in the 1970s. Looks like this. This is the total. So this is the total bending energy. This is an integral over the surface. This h here is the curvature. The h0 is a reference curvature, so the curvature that the cell would have if it's not forced by any external constraints. And this kappa b here is a bending modulus. The curvature can be written like this. So for an infinitely thin sheet inside in a 3D space, the curvature is the Laplacian, or the surface Laplacian, the so-called Laplace-Beltrami operator applied on the surface. And then if one derives from this energy, if one derives the forces, one needs to take derivatives. So that's what it looks like here. That's the delta F. That is the bending force density on the surface. And that has as the most important ingredient here that, again, this Laplace-Beltrami now applied on the curvature. So you're taking the second derivative of the curvature, and the curvature already is the second derivative. So what you have to do here is to take the fourth derivative on a discretized surface. And that is numerically, of course, as most of you will know, a very challenging issue. And there are, in fact, many different approaches out there how this can be done. And we just, a few years ago, we made a kind of detailed comparison between these different approaches and having the different advantages and disadvantages. So much for the cell. We're now going to look at the fluid solver, the Lattice-Boltzmann method that we're using. Lattice-Boltzmann is a method that does not start from the Navier-Stokes equations. So it ends up with the Navier-Stokes equations, but the Navier-Stokes is not the starting point. The starting point is, in fact, a picture of molecules. So we imagine molecules. And the difference then to MD simulations is that we restrict the positions of these molecules on a grid. So there's a grid, and the molecules can only sit on these grid points. They cannot be in between. The immediate consequence of that is that we're not only discretizing space, but we're also discretizing velocity space. Why is that? Because if a molecule is sitting on a grid point and you would allow for a continuum of velocities, at the next time step, it would not hit another grid point, so it would be off lattice, it would move away from the lattice, so you have to discretize the velocities, allow only such velocities which transport a molecule from one lattice site to a neighboring lattice site. So you're discretizing space and velocity space. We then don't work with individual molecules, unlike MD, but we work with molecule distributions. So we only count how many molecules on each grid point exist with a velocity in a particular direction, with a velocity to the upper left neighbor or to the lower right neighbor. That's the distribution. To relate this to macroscopic observables, like the density, one can then simply sum over these distributions. So they're normalized such that the sum gives you the local density. To get the velocity, you again sum over the distributions weighted with the velocities from one lattice site to another, and that gives you the macroscopic velocity, which then is a continuum. So the macroscopic velocity is a continuum because these particle distributions, they are continuous quantities. The time step in Lattice-Boltzmann consists of two parts. The first one is called the collision, and the second one is called the streaming. So collision is that the molecules on each lattice site, they interact, they collide, and they can exchange momentum. So the collision step, that is modeled by a relaxation to equilibrium. So given a macroscopic velocity on a given lattice site, you know that the molecules will have in equilibrium a Maxwell distribution. So you write down the Maxwell distribution, and then you take a relaxation term towards this Maxwell distribution. The parameter that you get here is a relaxation time, which is called tau. The streaming step then simply transfers. So you have the molecules with a velocity going to the upper right. All these molecules are transferred from one lattice site to the upper right neighboring lattice site. So it's a very simple very simple algorithm. To connect this to the Navier-Stokes, you can write down momentum conservation. So you can basically expand this collision operator, and that gives you a connection to the Navier-Stokes equation by matching the different moments, and that gives you the viscosity. So the viscosity, in fact, does not directly enter into lattice-Boltzmann. What does enter is the relaxation time tau, and the relaxation time tau is connected to the macroscopic viscosity. Some advantages and disadvantages. One advantage is that lattice-Boltzmann is very simple to code. That is one of the reasons why it's so popular. In fact, you can write down a full 3D lattice-Boltzmann solver in 100 lines of C++ code without any problem. So it's really a simple, very, very simple algorithm. It is very highly parallelizable, which means that it scales extremely well on GPUs. So it's very well suited to GPU computation because GPUs are built actually in the same spirit as the lattice-Boltzmann. They are built massively parallel, but very simple. It's the same as LBM. It's very simple, but it's massively parallel. Now, of course, it also has some disadvantages. One of the disadvantages is that it's a compressible method. So it's in fact because these densities, these particle distributions, they can change. So it's not strictly incompressible. The viscosity is linked to the grid parameters. If you remember the formula from the slide before, there was not only the tau, there was also a delta t and a delta x. So if you change your time step, that changes your viscosity. They're not fully independent. And that gives you a restricted range of stability, so you cannot take any parameters and simply throw it on a lattice-Boltzmann and it will work. Usually it does work, but not always. To give some typical numbers, the grid size that we're using is about 100 cubed up to 400 cubed, so that's kind of the standard grid sizes. It's parallelized here typically on 1 to 4 GPUs. And the number of time steps is fairly large, so it's quite common in lattice-Boltzmann simulations to have millions of time steps because the time steps are very small. So the physical time steps, the physical delta t, is in fact something like below a microsecond, or can go down to even nanosecond depending on viscosity. So it's very small time steps, but you can take many time steps because each time step is very simple. So let me just check the time a little bit. We need to couple that to the cells, to the cell mechanics. So the red blood cell model that I showed you before, that is done by a method called the immerse boundary. Immerse boundary, in fact, was invented long before lattice-Boltzmann, so it's not specifically for lattice-Boltzmann, but it works very nicely for lattice-Boltzmann. The idea is that you have the lattice-Boltzmann grid, that's the blue points here, and you have the membrane. The membrane vertices, so these triangles, they're off lattice. They're not confined to the lattice. They can be anywhere, but they need to speak to the lattice, and to speak to the lattice, we do interpolation, and we do spreading of forces. So that's the two directions. The first one is we have the fluid velocity on the grid points. We need to interpolate the fluid velocity on the position of the vertex to move the vertex. It's exactly no slip, so the vertex doesn't have a mass. It exactly moves with the local fluid velocity. And then the other way around is to take the force from the membrane, so these stretching and bending forces, and to include them as a source term into the collision operator, so into the Navier-Stokes equations. With this, I come to the second part. So we have now shown you the method that we're using. And I now want to show you how we apply this to the formation of blood platelets. Let me start by saying a few words about platelets. Platelets are very small cells. They're used to stop bleeding after injury. So if you scratch yourself, then it's the platelets that first respond, and they form a plug, and that kind of seals your vessel to stop bleeding. That's the function of platelets. To do this, they're activated. Before they're activated, they look like this. So they're like small ellipsoids, and that's the state that we're interested in. So we're not looking at the activation. Platelets are the second most abundant cell after red blood cells in blood flow. The lifespan is only 7 to 10 days. So it's not like you have a set of platelets and you keep them in your body until they're used, but they're produced and they're degraded again if they're not used, and most of them, in fact, are not used. And that is a problem because that restricts the duration of blood conservation. So if you donate blood, then you cannot store this forever. And one of the limits is this degradation of the blood platelets that you have donated. For that reason, people are trying to devise bioreactors, so microfluidic devices that can produce such platelets. And that is one of the motivations why we and others want to understand how this platelet formation actually works. To do that, we'll start with the in vivo situation. So I'll show you how platelet formation works in vivo. It happens in bones. So this is the bone. Inside the bone, there are cells called megakaryocytes. They're very large cells. And they're sitting right next to the blood vessels. So even in your bones, there's small blood vessels. That's the bloodstream up here. And what they do is they grow these protrusions, these extensions here into the bloodstream. These extensions grow. They're called the proplatelet. And then eventually these proplatelets, they rupture, they break up into pieces, and each of these pieces eventually becomes a platelet. This process is extremely efficient because these platelets only live for 7 to 10 days. Your body constantly needs to reproduce these platelets, and in fact, a healthy human body produces, on average, 1 million platelets every second. So all that we're sitting here, that we're speaking here, we're constantly producing 1 million of these platelets per second. So 1 million times this process is happening in all our bodies. So that's one of the fascinating things on this process, and what is a motivation to study that. To reconstitute this in vitro in bioreactors, there are different approaches. I'll show you two examples here. The first one is this. This is a microchannel. So the channel is up here, and there's a barrier in between, and that barrier has holes. And below here, there's a suspension of these megakaryocytes, and the megakaryocytes are getting trapped in these little holes. And then they grow these protrusions, these proplatelets. That's what you see here. That is the proplatelet here. And you see the bulges, these bulges. This thing will rupture, and each one of these bulges will eventually become a platelet. Another approach is to take micropillars. So it's a reactor that consists of micropillars, and you flow in a suspension of megakaryocytes. They attach to the micropillars, and then there's the flow, and that drags and that pulls these proplatelets out of the megakaryocytes, and then again they eventually rupture, and you're producing platelets. Now, the interesting thing about these in vitro setups is that you can regulate the velocity. In vivo, you cannot. The velocity, your blood flow velocity is as it is. Now, this you can regulate, and what people have found in these experiments is that the efficiency of the process extremely strongly depends on the flow velocity. If you add flow velocity, you can accelerate the production by up to 20 times. So that's not 20%, that's up to 20 times faster than without flow. One also finds that the size distribution, even though you are changing the velocity, is relatively robust. So what you get out is really functional platelets that have the correct size distribution, with the exception that sometimes you find platelets which have about twice the size as the usual size, the usual volume of the platelets. So what we have here is a system that has a very interesting coupling between active biology, so the biological process of the cell, which grows this proplatelet, there's really some active biology happening, and the surrounding flow. And these two speak to each other, and these two necessitate also each other, and that's what made it interesting for us to look at that. I'll show you the model that we're using. The model is in fact inspired by the red blood cell model that I showed you, so it's not completely new. We start with an extended proplatelet, so we're not modeling the growth. That's something that we're working on and that maybe eventually we can do, but for the moment, the proplatelet is already there, and we're focusing on the breakup. We're focusing on how this proplatelet breaks into pieces, into platelets. I said it's inspired by the red blood cell, so it has a membrane, a shell, and it has a liquid core inside. The membrane has the pieces that we know. It has a shear elasticity and an area conservation, so the Scalac law. It has a bending rigidity, that's the Helfrich flow, but it has now an additional contribution that the red blood cell does not have, and that's the active stress. So that is active processes done by actin contractility. So actin filaments are extended filaments, and there's motor proteins, myosins, so molecular motors that pull on these filaments. So you can imagine this like an isotropic random network, and there's connections between these network filaments, and there's motors which pull this network together. So that's the actomyosin cortex, and that has a contractile tangent. And of course, we have the external flow. The computational method, I'll do it very briefly because I showed you the one for the red blood cell before. So we need a model for the active stress. That is, this T here is the stress tensor. It's a surface stress tensor. That's why it has 2 by 2 components, because it lives on the surface. It does not live in 3D space. We discretize with vertices and triangles, just as the red blood cell membrane before. We need to get from these stresses to the forces. That's where for the bending case, that's where this Laplace-Beltrami popped up here. We're doing it a bit differently, but in essence, we have to take a derivative, a surface derivative of these specified stresses. We do this with a parabolic fit. The elastic forces are similar to the red blood cell, with shearing, area dilatation, and bending rigidity. And the fluid solver is lattice Boltzmann. For the results that I'm going to show you, we are using a package called Espresso, which has a lattice Boltzmann built in, which we extended. We are now transitioning, we now recently developed our own lattice Boltzmann code, so we're now transitioning to a different code, but things are still Espresso. I promise I'm going to talk a bit about the active stress, because that's the key ingredient that leads to the breakup. That's the key ingredient that distinguishes the platelet from the red blood cell. We need to do assumptions here, because in fact what these active stresses really are is not known. One knows that there's the actomyosin, and that the actomyosin is contractile, so one can reconstitute such actomyosin networks in vitro, and then one can see that they're contracting, so that is known. Contractile, we do it anisotropic. I'll speak about the motivation for doing that in a second. And we assume it to be constant in time and space. So the stress tensor, the active stress tensor that we're imposing looks like this. It has 2 components here in the axial direction, so Z in this direction, and in the azimuthal direction, so in the angular direction, and they can be different. The values that we're using, this is the value for the active stress. This is the anisotropy ratio. So it's a bit more the stress in the azimuthal direction is a bit larger than in the other direction. And for kappa S and kappa B, we use the red blood cell values, simply because for the proplatelets, they have not been measured, so nobody knows really what these values are, but mechanically they should be quite similar to the red blood cell. The radius is about 1.5 micrometers. So the main questions that we're asking is, what is the mechanism for this break? So why do these break up at all? What is the reason behind that? Can this model reproduce accelerated breakup in flow? So if we add a flow, do we see this effect that the breakup happens faster? That is seen in the experiment. And finally, can this mechanism also explain the occurrence of these double-sized platelets, these so-called pre-platelets? We'll start with a very simple case, platelet formation without flow. So this is the starting situation. This is the extended proplatelet, and we run the simulation. Because of this active stress, because of this contractile stress, the membrane contracts. So the tip here is pulled to the left, and it forms these bulges, and it forms these instabilities. We study this using a so-called chymograph. That's this thing here. So it has as axis the x-axis, so that is the axis along the proplatelet here. It has time. It starts up here, and then time runs downward, and the color coding is the radius. So initially, it's all the same color, because initially the radius is the same everywhere, and then we get these stripe patterns. So here, these are the bulges, and the light parts in between. This is where we get the holes, so to speak. From that, one can extract the wavelength of this instability. One finds a wavelength of about 14 micrometer, and one can take the relative wavelength, so the wavelength divided by the radius, and one finds a ratio of 9.3. And I guess that most of you in the audience have already seen this value of about 9, because that's exactly the Rayleigh plateau prediction. So that's exactly what you would expect for a liquid jet that breaks up due to surface tension. And in fact, if you write down the stress for a surface tension mathematically, it looks the same as the active stress that we're specifying, except for this anisotropy. So in principle, this is not very surprising that you would get the Rayleigh plateau instability for these computations. Now, of course, you want to check with reality. Does that make sense? Could that be realistic? So in nature, the proplatelet radius is about 1 to 2 micrometer. That would give a Rayleigh plateau prediction of between 28 and 230 micrometer cubed for the volume of these platelets. And that platelet volume, that has been measured, and one finds a distribution which starts at 15 for the very small ones and goes up to roughly 80 micrometer cubed. So that fits very nicely in that range. So that Rayleigh plateau picture is compatible with what is seen in nature. So we think that some Rayleigh plateau-like instability, and that's the hypothesis, is behind the formation of these platelets. We now do this in flow. We start with a homogeneous flow. So there's a homogeneous flow about 1 millimeter per second. That's the typical velocity that you have in small microcapillaries, so in small blood vessels where platelet formation happens. And that changes the picture in such that we're pulling on the proplatelet and we're pulling on these bulges, and these bulges start to move, but they still form. Here's the chymograph. You see the chymograph now slightly moves here to the right. That's because the flow is dragging, is pulling on the proplatelet. And here I'm showing you the time that it takes to form these platelets. So that's the breakup time. We start as soon as we can identify one of the platelets. That's in fact this one here. So as soon as we can identify, and then until it has finally formed. So the radius as function of time. And we do this here. This is for 0 velocity, and then we increase the flow velocity. And we see that the curve becomes steeper, so the formation becomes faster, and we increase the velocity, and it becomes still steeper, and we increase the velocity again, and in the end it becomes very steep. So in the end, this breakup happens really quickly. So in fact, this Rayleigh plateau mechanism, these simulations, can also explain the accelerated breakup due to flow that is seen in the experiments. Here's for the different platelets, so the different of these bulges, so the platelet 1, and here the platelet 2, and then eventually the platelet 3, the time as a function of the velocity, and you can see that it does go down. It doesn't go down by a factor of 20, so we don't fully explain this acceleration. The reason is that we're only modeling the second half of the process. We're only modeling the breakup. In fact, what also accelerates is the growth. So the growth of this proplatelet is also accelerated by flow, and that is not included in the model. So that's why we don't get the factor 20, but we do get a significant acceleration due to flow. We check the volume. The volume does not change much, so that is compatible again with the experimental observation. Right, so accelerated breakup, yes, our model can reproduce that. Now, in these blood vessels, you don't have a simple homogeneous flow, but you have a more complex flow, and we're now building up this complex flow by parts. So homogeneous was the first one, and the second one is a pure shear flow. So we fix the proplatelet here, and then we apply a pure shear flow. Again, the bulges form, so again, the instability happens. But now there's a new effect. If you focus on these two here, so you see they merge. So there's two bulges, and because of the shear flow, because the shear flow is dragging, these two bulges actually merge. You can see this also very nicely in the chymograph. They form as separate bulges, then they grow, and here eventually they merge, and then they form one large bulge, which then has twice the volume as the original one. So we hypothesize that the occurrence of these pre-platelets, these double-sized platelets, is actually due to fusion of these bulges, which is induced by the flow. So the flow is responsible for merging two of these bulges, and that gives you the pre-platelets. Now, in the real situation, you have a Poiseuille flow, so you have both. You have, it's like a combination of a homogeneous and a shear flow. We also study that. That's what it looks like. Bulges form. Strings are extended, so the bulges are pulled apart by the flow. And if you compare, let me see if I can stop this. Okay, I cannot pause the video, but if you look at the final state of the video and you compare it to this experimental image, you see that they really very nicely agree visually. So we think that this Rayleigh plateau really is an explanation for that. And we also check the other quantities. In the Poiseuille flow, we get merging, so we get double-sized pre-platelets. We get acceleration by flow. This is zero velocity, then the velocity increases, so it becomes steeper. Here the formation time goes down. The volume is constant except for this jump here at very high velocities, but roughly conserved. So to conclude this part, we think that this Rayleigh plateau mechanism, although there's no surface tension anywhere, it's a purely biological system. There's no surface, but there is an interface, and we think that this active stress that is created by the cell, by the ectomyosin cortex, plays the same role as the surface tension would do for a liquid jet, and therefore you get the same kind of instabilities, and that explains why these platelets form. Now to come to the third and last part about the anisotropic Rayleigh plateau instability. When I introduced the proplatelets, I showed you that we are imposing an anisotropic stress. So unlike surface tension is the same in every direction. So here we're using the axial and the azimuthal direction is different. So the motivation for that is the structure of cell membranes, because cell membranes often have anisotropic components. Here are two examples. The first one is what's called stress fibers. They are bundles of these actin filaments. So these actin filaments, usually they're oriented isotropically, the same in each direction, but sometimes, depending on boundary conditions, they can form these bundles, and then you have some elongated structures and they are aligned. So that breaks the symmetry. In the platelets, it's a bit different. In the platelets, the actin network is isotropic, contractile, but there's a second component that's called microtubules. Microtubules are relatively rigid structures. They're also lengthy, they're extended structures, and they can orient, and in fact, in these long cylindrical proplatelets, they are oriented along the axial direction, and there's a different kind of motor proteins that pushes on these, that makes them slide. So you have, if these two are two of these microtubules, they're oriented like this, and then there's a motor which pushes them. And that is what is currently thought to be the main mechanism driving this extension. And we include this by the anisotropic stress. So basically the idea is we have the ectomyosin cortex that is contractile isotropic in each direction, but then in one direction, along the axial direction of the platelet, you have the microtubules which are pushing, and that reduces the contractile stress from the actin. So you get a stress which is larger in the azimuthal, in the angular direction, than it is in the axial one, because of this combination of actin and microtubules. So what we do is we do the classical Rayleigh plateau approach. So we take an infinitely long cylinder and we impose a perturbation. And the difference is that we have now these different stresses, the gamma phi in the azimuthal direction and the gamma z in the axial direction. One can in fact repeat the Rayleigh plateau analysis in the same way as it was done originally, so there's no mathematical, no completely new obstacles happening, so that's why I'm skipping this. So the way is the same. You do linear stability analysis, and you get in the end a dispersion relation, so omega squared. And you get a new term here, and that new term has the ratio of the anisotropy. So that's what you do. So you don't get this term if you have an isotropic case. Then you look at the stability. So whenever omega squared is positive, the system is unstable. So that is the wave number. That is omega squared, and that's the unstable region. Here for the isotropic case, the boundary is 1. So the critical wave number is just the radius. And if you have a situation where the azimuthal stress is larger, so you're more contracting, you're shifting this stability region, you're extending the stability region, the unstable region, sorry, you're extending the unstable region, so you're destabilizing the system, so you get wavelengths which in the isotropic case would be stable, now they're not stable anymore. And the opposite, if you take the axial stress larger than the azimuthal stress, then the stability region the stability region grows. So it's a stabilizing effect. The next step after having the dispersion relation is to look at the dominant wavelength. So what is the wavelength out of all these unstable modes? Which mode grows fastest? Here I've shown this fastest growing mode, lambda, as a function of the anisotropy ratio. So 1 here, that's the classical Rayleigh plateau. If you go up here, you see you get here the 9.3, so you get the classical Rayleigh plateau value. And if you are in the destabilized case, so where the azimuthal direction, the azimuthal stress is larger than the axial stress, the wavelength shrinks, so the dominant wavelength becomes shorter. Whereas if you go towards the stabilized case, then the dominant wavelength increases. We check this by simulation, so we do lattice-Boltzmann simulations on this situation, and we just look at the breakup, and we see which mode grows fastest, and that is in nice agreement with this theory. Now, the classical Rayleigh plateau is for the inertial case. If you look at these proplatelets, there's no inertia anywhere. In fact, in most of these microscopic bioflows, inertia is completely irrelevant. So you're deep, deep in the low Reynolds-Stokes regime. So we repeat this analysis for the Stokes case. Rayleigh plateau for Stokes flow has been done before a few times. We took the analysis from this paper because there we found it to be the most readable. And again, we extend it to anisotropic stresses. So again, we put these different stresses and we repeat the analysis. And we find a growth rate that plays the same role as the omega squared in the other case. And that again has this term here. So this term here pops up again, the anisotropy ratio times the square of the radius with the wave number. The picture does not look qualitatively different. So the black curve now is the theory for Stokes flow. It's a bit higher. The dominant wavelength is a bit higher, but overall it also grows with the anisotropy ratio, and we can also verify this with simulations. Now that is a very different kind of simulation. That is boundary integral. So we have these two methods. One is lattice-Boltzmann. That's what I focused on today. But there's also a viscous boundary integral method, and that is then used for that case and is in nice agreement with the theory here. So the dominant wavelength depends on the anisotropic stress. The very final ingredient is bending. So you can add bending to this analysis. You can also add shear elasticity, but I'm not showing that. And here is the curve. The red curve is without bending, and these squares here are with some bending. So we define a non-dimensional bending strength. And for the isotropic case, that has been done before in these two papers. And what these people concluded is that bending is not very important. And in fact, if you increase the bending, so 0.1, 0.5, 0.1, you see that for the isotropic case, indeed, bending is not very important. So bending stresses don't really change the Rayleigh plateau mechanism. But as soon as you go to the anisotropic case, like here or here, you see that there's quite a significant effect of bending. So bending and anisotropy here really talk to each other. So bending becomes important when you have anisotropy. Now let me come to the conclusions. What I showed you was the lattice-Boltzmann method for fluid flow. I showed you the pros and cons, and I showed you the basic idea of having these fluid particle distributions. The computation method for active stresses on these membranes embedded in a 3D space, non-axisymmetric, so fully three-dimensional, so that we can do shear flow and Poiseuille flow. I showed you the hypothesis that we think Rayleigh-Plateau mechanism of a different kind, not surface tension, driven by this active stress, is behind the formation of blood platelets. And finally, I showed you how the Rayleigh-Plateau analysis can be analytically extended to the anisotropic case. And some acknowledgements. This is the group picture, which is a bit outdated. So Katharina Bresel, she did lots of the work, and Stephan Gekle, who has finished, so he's not on that picture anymore. So that's the two people that did most of the work here. And funding comes from these different sources. Thank you very much.
Thank you very much, Stephan.
Hello, so you showed some results for filaments that were kind of a cylinder and then breaking up in flow, but presumably the flow is always there and it's slowly growing and breaking up as it goes. Does that change? Do you know what I mean? Sorry, so you somehow had a uniform cylinder that hadn't broken up at all, and then you sort of turn on the flow.
Yeah.
But actually, the flow is always there, and this thing is continuously growing and breaking up and...
Probably yes. Probably that's the reason why we do see acceleration, but not by such a drastic factor as the experiment.
Okay.
Because in reality, the flow also affects the growth, and that's what we're not modeling.
Yeah.
So yes, that will change.
At the late stages of the instability, are you able to see satellite droplets, or do you see long filaments like what we have for polymer solutions?
One can. We also saw satellites, yes.
Okay.
Not in the analytical theory, of course, but in the simulations, one can also see satellites. The anisotropy does affect satellite formation, but not in so we couldn't make any intuitive conclusion on how it does. So you change the anisotropy and the satellites change. Yes.
But there are satellites.
There are satellites, yes.
So how do you get the receptors on synthetically, and does speeding it up mess up the receptor distribution?
The receptor distribution inside
On the surface of the platelets, yeah.
We don't model them because
but what happens in practice?
In practice, they're not relevant for the formation of the platelets. Receptors become important when they attach, so not when they're constructed. So we're focusing on how these platelets are being constructed from the large progenitor cell, from the megakaryocyte.
But how do the receptors get there then?
They are already, so they are produced so the megakaryocyte has a nucleus, so it can synthesize.
But it's synthetically, right?
Yes, but the synthetic case needs the stem cell. So you need the stem cell. You cannot produce them out of the blue. They're just way too common. They have hundreds of proteins.
Sure.
So you need the stem cell, and the stem cell produces the receptors. And during the growth of the proplatelets, the receptors grow along, so they're already there inside the proplatelet, and then it ruptures, and then they just stay there in the platelet.
Thank you.
And they're needed when it's attached.
You carefully avoided mentioning the word elastic. You said...
Sorry. You have to.
Okay, that was a very stimulating presentation, but you carefully avoided mentioning the word elastic. You said anisotropy and so on and so forth. What do you mean by anisotropy? The stresses and elasticity are anisotropic, yet the properties, let's say, the area dilatation modulus, it's isotropic. Are you referring to a difference between the axial and azimuthal area dilatation modulus?
Yeah.
Am I getting it wrong?
No, I think you are getting it right. So we're adding. So we have the passive elasticity, If I avoided that was not on purpose because it is important. So we have the passive elasticity, that's the shear elasticity and the bending, for example. And that we model as isotropic. And then on top, there's the active stress. And the active stress is not elastic. The active stress is not connected to deformation. So it simply comes on top. It's a stress that comes on top of the elastic stresses. But the elastic stresses are there, and they are isotropic. The active stress is anisotropic.
So then, where does it come from, the active stress again?
From the molecular motors.
From the molecular.
Yeah, that's the molecular motors. Yeah.
Just to clarify, you say it's an active stress, but actually it's just their constant, so you could think of it as an anisotropic pre-stress.
You could think of that as well.
You don't yet have
Absolutely.
activity in your model coupled to the biology.
Yeah.
Thanks.
There's a lot more that you can think about.
Yeah.
For example, these myosins, these molecular motors, they will not be distributed exactly like that. So they will diffuse, and so it's the starting point.
Okay, thank you very much, Stephan. Thank you very much for your very nice.